Bismillahirrahmanirrahim. Today we will discuss about the Sanger DNA sequencing process. So Sanger DNA sequencing process are also known as dideoxy DNA sequencing or chain termination sequencing. So uh, first of all, we define that what is DNA sequencing. So DNA sequencing is basically a process in which we can find out the order of nucleotide sequence. So let's go to start the topic. So first of all, we have to know that what types of constituent are used in the Sanger sequencing method. So these constituent are, the first one is a template strand of DNA. The second one is a primer. The third one is a DNTPs, and fourth one is DDNTP. DD stand for dideoxynucleotide triphosphate, and buffer solution are also required. And the last one is take take polymerase. So these all are the constituent part single DNA sequencing. So a question is arise that why Sanger sequencing DNA are also known as dideoxy nucleotide sequencing process, because we modified a, a nucleotide sequence. Normally in a nucleotide, this is the structure of a nucleotide. In normal nucleotide, the in the DNA, the second hydroxyl group are present on second carbon position and also present in the third carbon position. But in dideoxy, the third carbon, carbon miss the oxygen group. We remove the oxygen from the third number of carbon. So when the process of PCR are started, so when this dideoxy nucleotide are came, so the process are terminate. So is that why this process are also known as chain termination process. Now let's go to start the mechanism of this process. And the mechanism, we have to take four different types of tube, and these tubes, all the constituent of PCR are present. So the difference between normal PCR and the Sanger sequencing PCR is that in the Sanger sequencing PCR, the extra one dideoxy nucleotide are present in all tube. And this tube, the dideoxy GTP are present, and this tube dideoxy CTP are present, and this tube dideoxy ATP and dideoxy TTP are present. So when we start the PCR and when we add our template into these four different type of tubes, so what happened? Now the DNA polymerase, the take polymerase add nucleotide into a template strand. This is our unknown template strand in which we want to find the order of nucleotide sequence in this template strand. So in normal PCR, the dideoxy are absent, and here the dideoxy nucleotide triphosphate are present. When we add all the constituent as well as the dideoxy GTP, so they this fragments are come here. When the polymerization process are started, so take polymerase add C against to G. We know that the complementary of G is C. So in this tube we add DDGTP. Now in this C position the chain will be terminated because the dideoxynucleotide are attached. So when this are attached, the termination are takes place because in the third carbon position the oxygen are absent in this dideoxy-GTP. So the polymerization are not takes place. Now in the another here the all and other C are present. So this the take polymerase add these complementary nucleotide again this, but when the C are occur, so this G are attached to that which are modified nucleotide. So two fragments are come here in this first PCR tube. Now let's go to come the second PCR tube. It consists all constituents of PCR and as well as the dideoxy CTP are also present in this tube. Now these three fragments are made in this tube. The first one is C. We know that here C are present, so we know that the complementary part C nucleotide is G. So this when the modified nucleotide are come here and attached to this, so this are terminated on this fragment. Now the second one GG are also present in this unknown sequence. So here DNA polymerase add the complementary nucleotide to this unknown sequence. So here when the G position takes place, so the modified nucleotide are came and attached to its complementary, so the chain will be terminated. The implication of the sequence are terminate. Now here the other C are here the other C are present. Now in this tube C are also present when another G are also present when DNA polymerase 8 its complementary, and when come on this G, so here attach the modified C nucleotides. Now there are three fragments are formed in this C tube which are modified. Now in the third PCR tube we add dideoxy ATP which are complementary to T nucleotide but that are modified, and we remove the oxygen from the third carbon position in nucleotides. So here we see that in this strand only one A are present, only one T are present. So per A complementation there are the T will be attached, so here are T are present. So now when the amplification of this strand are started in this third tube, so here the A are the T the and this T the A are attached and this chain will be terminated. Now in last tube the GTTP and all PCR constituent are present. When the amplification of this strand are occur and when the A nucleotide are takes place, so the DNA polymerase enzyme add the abnormal DNA nucleotide, and here the termination are occurred because they add the dideoxy triphosphate. And we know that the other amplification are cannot be occurred because here the dideoxy TTP are attached. Now we take these all tube and run on the gel electrophoresis. So here this is the DDGTP, this tube tray, and this is the another, and this is the other gel tray per separate tube. We add our sample and the gel, and we run that. So for the DGTP, there are two fragments are formed. One is 2 nucleotide and the another is 7 nucleotide. We write the numbers here, and at its front we draw the line to represent the fragments. So here two um, fragments are occurred, so we draw it into a specific position. Now also we write this, these are in the gel tray. So in the first uh, uh, fragment are DCTP. So here we write C, and the another is a G, which are present in this position 2. And then we write the third, which are present basically A, and so on. So here we take the complementary of our this sequence. So the complementary of this sequence is G C T G G A C. Now we match this sequence which are come from our Sanger-Sequenzierung process to this unknown sequence. Now here G, and here also G present, C C T T G G G G A C. So we done the sequencing of unknown template strand. So hopefully you understand this lecture. Inshallah, see you in the next time. Thank you.